Assalamualaikum Prof. Dr. Sarah. Assalamualaikum.
Waalaikumsalam. Assalamualaikum semua.
Salam. Prof, unmute.
Okay, tolong check ni background okey ke you all tengok semua?
Okay.
Okey. Pasal saya ni nampak terbalik lah kan. Jadi saya pun teringat
Betul.
terbalik. Okey, alright.
Assalamualaikum. Semua apa khabar?
Baik. Okey. Waalaikumsalam.
So you're going to start about a few minutes more. We wait for our other friends to come in first. Alright. Okay. So now it's ready to past 10. So I think we can start our webinar today. So Assalamualaikum and good morning to our honourable guest, Professor Dr. Abdul Rahman Milan, President of the Persatuan Genetik Malaysia, PGM. Welcome, Prof. Professor Dr. Yamin Yassin.
Okay, thank you.
Professor Dr. Yamin Yassin, Rektor UiTM Cawangan Negeri Sembilan, Dr. Siti Sarah Ibrahim, Deputy Rector of Research Industry Network, Community and Alumni Division UiTM Cawangan Negeri Sembilan, Dr. Ida Muriani Mat Yassin, Head of School of Biology, Dr. Nur Aisyah Hassan, the Chairman of BioMECs, our distinguished speakers today, Dr. Amirul Adli Abdul Aziz from UiTM Cawangan Negeri Sembilan, and Dr. Muhammad Asyraf Hatta from University Putra Malaysia, respected Persatuan Genetik Malaysia Exco members and respected participants. So before we begin, let me introduce myself. I'm Nurhamimah from UiTM Cawangan Negeri Sembilan, and I will be responsible for hosting our webinar today with the title Beginner's Guide to CRISPR-Cas9. So for your information, it's organized by special interest group named as BioMECs, which is an acronym for Biotechnology, Microbiology and Environment Collaborative Sciences. So BioMECs has been established on the 1st of April 2021, just recently. So our vision is to be an organization oriented to research, innovation and multidisciplinary collaboration to meet science and technology needs in the future. So with our motto, Navigating Biology, BioMECs conducts research that integrates molecular techniques involving expertise in genetics, molecular biology, microbial physiology, microbial genomic, biosensor, immunology, structural biology, and environmental biotechnology. So don't forget to like our Facebook page as shown in the screen. So make sure you get the right logo, BioMECs UiTM, our Instagram page. So this is our Instagram page, our YouTube channel, and also our website. So that is our website. So our webinar today, Beginner's Guide to CRISPR-Cas9, is our first webinar organized by BioMECs. So on behalf of the organizers, I would like to extend my warm welcome to our guests and all the participants here in Google Meet, as well as those watchers from Facebook live streaming. And thank you for making efforts to join our webinar today. So without further ado, I would like to invite Dr. Siti Sarah Ibrahim, the Deputy Rector of Research Industry Network, Committee and Alumni Division, UiTM Cawangan Negeri Sembilan, to deliver her welcome speech.
Bismillahirrahmanirrahim. Thank you to our moderator. Assalamualaikum and a very good morning to Yang Berbahagia Professor Dr. Abdul Rahman Milan, President of the Persatuan Genetik Malaysia, PGM, Professor Dr. Yamin Yassin, Rektor of UiTM Cawangan Negeri Sembilan, Yang Berusaha Dr. Ida Muriani binti Mat Yassin, Head of School of Biology, Dr. Nur Aisyah Hassan, the Chairman of BioMECs, our distinguished speakers for today, Dr. Amirul Adli from UiTM Cawangan Negeri Sembilan, Kampus Kuala Pilah, and Dr. Muhammad Asyraf from University Putra Malaysia, respected Persatuan Genetik Malaysia, ESCO members and also the respected participants. Assalamualaikum again. It is a great pleasure for me to welcome you to the first webinar organized by the SIG Group's BioMECs with the title The Beginner's Guide to CRISPR-Cas9 in collaboration with Persatuan Genetik Malaysia. Despite the unprecedented situation with the global COVID-19 pandemic that does not allow us to gather physically, this webinar is held virtually using Google Meet platform and the live streaming of BioMECs Facebook. I would like to thank to all the distinguished guests, panelists, moderator and participants for taking a time to join from near or far in today's webinar. Ladies and gentlemen, Research Industry Network Community and Alumni Division, PGIMA UiTM Cawangan Negeri Sembilan, is entrusted to assist academicians in the research developments, collaborating with government agencies, private organizations and communities to enhance graduates' marketability as well as to generate research opportunities in all three campuses in Kuala Pilah, Seremban and Rembau. One of our initiative is establishing scientific interest group, which is SIG, to accelerate more research programs among staffs starting last year. There are several groups has been registered under SIG at UiTM Cawangan Negeri Sembilan, including BioMECs. So I would like to take this opportunity to congratulate BioMECs on the establishment of this group, and I'm looking forward to see many efforts to flourish their research development in here. As everyone is aware, today's title is about CRISPR-Cas9. CRISPR-Cas9 is a genome engineering technology that enables scientists to easily and precisely edit the DNA of any genome. CRISPR is quickly transforming the world around us by revolutionizing our approach to scientific research. The application of this CRISPR-Cas9 technology is limitless, including the application in diagnostics, preventing diseases, gene therapies, and improving crops. On behalf of Industrial Linkage Community and Alumni PJIMA UiTM Cawangan Negeri Sembilan, we will give our full support to encourage more webinars like today to expose more people in the new developments and latest finding in these scientific areas. Again, I would like to congratulate School of Biology, UiTM Cawangan Negeri Sembilan, especially to the SIG BioMECs committee members, which is led by Dr. Nur Hassan, for their dedication and efforts to organize this engaging webinar. Hope it will give an inspiration to other SIG outside. On behalf of UiTM Cawangan Negeri Sembilan, I would like to extend my sincere appreciation to Persatuan Genetik Malaysia for their enormous support to make this webinar possible. I wish the webinar great success. Thank you, and again, congratulations.
So thank you, Dr. Siti Sarah, for the remarkable speech. So next, I would like to invite Professor Dr. Abdul Rahman Milan, the President of Persatuan Genetik Malaysia, to deliver his opening speech and also to officiate our webinar today.
Thank you, Dr. Nurhamimah. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi taala wabarakatuh and salam sejahtera. Yang berbahagia Dr. Siti Sarah Ibrahim, Deputy Director Research Industrial Linkage Community and Alumni UiTM. Yang berusaha Dr. Ida Muriani Mak Yasin, Head of School of Biology, Dr. Nur Hassan, Leaders of Special Interest Group BioMECs, Dr. Nurhamimah, Zainal Abidin, Chair of SIG Webinar Series in collaboration with PGM, distinguished speakers, respected PGM ESCOs, and all participants. On behalf of the Genetic Society of Malaysia, I would like to welcome all participants to the SIG Webinar Series in collaboration with PGM. We are indeed very pleased that PGM seminar continue to take place, updating scientific community and public on the issue related to genetics, especially the current situation due to COVID-19 pandemic. I feel we are getting used to the new norms where most events are held virtually. I am very delighted that we have received an overwhelming numbers of local and international participants, and this shows that the importance of genetic approach to many researchers. The society is very much indebted to School of Biology UiTM, especially to the BioMECs, a special interest group, for agreeing to co-organize the event. My highest appreciation goes to the members of the BioMECs under the leadership of Dr. Nur Hassan, as well as the PGM ESCOs who have been working very hard to make this day a reality. Ladies and gentlemen, CRISPR-Cas9 is a genome editing tool that is creating a buzz in the science world. It is faster, cheaper, and more accurate than previous technique of editing DNA and has a wide range of potential applications. This free webinar brings this in two enthusiastic speakers who will be talking on CRISPR-Cas9, including the application of this technology in the plant field. I hope this webinar will provide yet another opportunity for members and non-members alike to touch base with the developments and latest findings in this area. Dear all participants, I encourage everyone to visit our PGM website, www.pgm-my.org, for more information about PGM and its activities. Kindly register as a member for the unlimited accessibility to the website and to enjoy members' privilege. Especially we have coming a new event, the big event, International Plant Breedings on the March 2020. I wish to convey my heartiest gratitude to all invited speakers for accepting our invitation to speak and share your research and finding here with us. To all participants, members, and non-members, thank you for choosing to spend your time with us today. I hope all participants will find the seminar very useful, informative, and enlightening. With that, dengan lafaz Bismillahirrahmanirrahim, I declare SIG webinar series in collaboration with PGM is officiated. Thank you. Bismillahirrahmanirrahim. Wassalamualaikum warahmatullahi wabarakatuh, and thank you very much.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Prof. Rahman, for the speech. So, ladies and gentlemen, so this is our first webinar. So, our team has prepared a special montage to introduce you about BioMECs. So, let's play the montage now. So I hope you enjoy the montage prepared by our team. So before I introduce our distinguished panels today, so I would like to ask everyone in the Google Meet to open the camera for our photography section. So I hope the participants in the Google Meet can open the camera so we can take a photo together. So it's ready? So, all right. Dr. Aisha, okay.
Okay.
Okay. Everyone is in the photo.
Okay.
All right. Okay, thank you, everyone. So, okay. Today, our webinar will be divided into two sections as there are two amazing speakers with us today. So the question and answer section, the Q&A, will be after both speakers' talks, and participants can drop your questions in the Slido link given in the chat box for those in the Google Meet, as well as in the Facebook comments, those who watch us live streaming. Or you can scan QR code that will be displayed on the screen later at the end of the both sections. And later, then I will attend your questions. Lastly, the link for the attendance and e-certificate will be given at the end of the webinar. So ladies and gentlemen, the talk for the first section will be given by Dr. Amirul Adli Abdul Aziz, who is a senior lecturer in the Faculty of Applied Science, UiTM Kuala Lumpur. Dr. Adli obtained his degree in Bachelor of Science in Biochemistry from the University of Sheffield, United Kingdom. So Dr. Adli then pursued PhD in Structural Biology in the same university and graduated in 2018. After graduation, Dr. Adli worked in Institute of Medical Research, IMR, where he attempted to use CRISPR-Cas9 in his project. So without further ado, I would like to invite our first panel, Dr. Amirul Adli, to give his talk on the basic theories and knowledge on CRISPR-Cas9. So now the floor is yours, Doctor.
Okay, thank you, Madam Moderator, Dr. Nurhamimah. Assalamualaikum warahmatullahi wabarakatuh, and a very good morning, rectors, deputy rectors, professors, doctors, and my fellow audience. I am very honored to be invited to speak in this event. And being a member of BioMECs, I'm proud to share with you today a little bit about what I know, which is the CRISPR-Cas9 system as a genomic editing tool. Let me share my slide first. Can I check if everyone can see the slide?
Yes, yes.
Okay. For those who are on Facebook Live, which is probably the majority of you, I'll just hang on a bit and see if my slide is displayed there, and I can see it's displayed quite well there. Okay. So if at any point my slide is not presenting or my audio is missing or breaking, please let us know by the Facebook comments or the Google Meet chat. Okay. So without further ado, let's get to it. Okay. CRISPR-Cas9 as a powerful genome editing tool. It is quite a fascinating technique to edit genome, and it's only recently recognized as one of the powerful editing tool that has been applied in many different fields. Okay. Before I begin my talk, let me just reiterate that the title of our webinar today is Beginner's Guide to CRISPR-Cas9. So the rest of my talk today is especially tailored to address beginners who are only starting to get involved or are interested in trying out CRISPR-Cas9. It is not meant for advanced CRISPR-Cas9 user. If you are having difficulties in your experiments, this talk is probably not tailored for that purpose, because we want to the purpose of BioMECs is to enhance research network and to kind of invite and introduce everyone to the the most recent tools. So we aim specifically for beginners, so that more people are getting involved in this really interesting technique. Okay. Right. So let's talk about the origin, how it started in nature. Okay. It's really very simple. Okay. It involves two simple microorganisms. Okay. This is a bacteria. Okay. It doesn't have to be a specific bacteria, but the CRISPR-Cas9 system is utilized by a bacteria called Streptococcus pyogenes. There are many different CRISPR systems that use different Cas enzyme, but we are, just for simplicity, I'm just going to explain to you about the CRISPR-Cas9 system utilized by Streptococcus pyogenes. And this is a bacteriophage. Okay. Bacteriophage is a virus. Okay, the phage, the term phage here corresponds to a virus, and bacteriophage is a virus that infects bacteria. Okay, it is not a bacteria; it is a virus that infects bacteria. So don't get confused with the term bacteriophage. Okay, so how does it infect bacteria? Okay, unlike maybe most of you have already started getting into virology, judging by the pandemic, the global pandemic that we're facing right now. COVID virus is a coronavirus, so it's a spherical type virus, and there are many different types of viruses with many different mechanisms of infection. In this talk, we are going to look at how bacteriophage infect bacteria. Okay, so this is a typical structure of a bacteriophage. It has a head there, or what we call capsid, inside which it has DNA or genomic material for the virus. There's a sheath there and a base plate. Okay, so we are going to look into the infection mechanism of a virus. Okay, the so-called lytic cycle. Okay, bacteriophage infection has two stages or two different mechanisms. You have a lytic cycle and a lysogenic cycle. But in terms of CRISPR-Cas9, we are going to look into the lytic cycle, which is where the bacteriophage injects the DNA inside the bacterial cell, and then it hijacks the bacterial mechanism or bacterial machinery, and then the virus replicates in the bacterial cell, and then you have assembly of the viruses and then releases of new viral particles, and this often results in the death of the host cell, which is in this case the Streptococcus pyogenes cell, and hence it is called the lytic cycle, which is lysis or cell death or cell breakage. Okay, now of course the primary aim of the virus is just to replicate and to produce more viruses, because that's what viruses do. They don't think of anything else. They just infect and replicates. Okay, in terms of bacteriophage, sometimes they go into the lysogeny cycle, where they can just integrate the genome and stay dormant in the host cell. But when the time comes, they replicate and produce new viral particles, and that results in the death of bacterial cells or the host cells. Then of course, with that relationship between the bacteriophage and the bacteria, the sole purpose of the bacteriophage is to duplicate, and they are able to fulfill their purposes, and in result to that, the bacteria is lysed or is facing eternal doom. So through time, through evolution, the bacteria has managed to evolve a system, a defense mechanism to fight off a viral infection, and that is what CRISPR-Cas9 system is, or generally CRISPR system. Let's look into the basics. I hope you are not intimidated by all these diagrams. You know, it looks like a downstream madness, but it's really very simple. Okay. First of all, as I said earlier, the virus injects the viral genome or the viral DNA into the bacterial cell, and then the CRISPR system has evolved in which the target gene of the viral DNA with the requirement of this PAM sequence. So PAM is a short term for protospacer adjacent motif, which is in short called PAM because it's easier. And it's simply just a sequence of DNA bases. In the CRISPR-Cas9 system, it's NGG. So for those of you who are not familiar with DNA, it's a deoxyribonucleic acids of nucleic acid monomers. There are 4 bases, A, T, C, and G. I'm not going to explain what each one is, but for a PAM motif of PAM sequence required for the CRISPR-Cas9 system is simply NGG, where N corresponds to any basis. It can be AGG, it can be TGG, it can be CGG, and it can also be GGG. So this target gene of the viral DNA that has this PAM sequence will first be incorporated into the CRISPR loci. Okay, so a loci in the genome, in the bacterial genome, is just a place or a location where a cluster of genes are located, and all those genes are involved in the same system, and that is why we call it the CRISPR loci. So all those genes have functions to fulfill the overall CRISPR mechanism. There are several genes that are involved there. The target DNA, the target viral DNA will be incorporated here, surrounded by the CRISPR RNA sequence. Okay, so this CRRNA is the CRISPR RNA sequences. Oh, by the way, CRISPR is actually short for clustered regularly interspaced palindromic repeats, short palindromic repeats. So it's quite a long word, but those scientists have come up with a really interesting name to remember it by. So you don't really have to memorize the long term. So CRISPR is the short term that everyone is familiar now. And then there are also several genes that involve in the acquisition of target viral DNA into the CRISPR loci. And then you have this tracer or trans-activating CRISPR RNA sequence, and also a Cas9 sequence. So Cas9 is a sequence that code for a nuclease. Nuclease is an enzyme that targets nucleic acid or DNA. And Cas9 specifically is an endonuclease. So endonuclease is the enzyme that targets DNA sequence in the middle. It doesn't have to cut from the outside of or from the ends. It can target anywhere in the middle of a double-stranded DNA. So that is what an endonuclease is. So in essence, this CRISPR system incorporates the target viral DNA into the sequence, and then they produce three important players in the CRISPR defense mechanism in a stage where we call cRNA biogenesis, whereby what you get are this tracer RNA, which is a short RNA sequence, and cRNA, CRISPR RNA, containing the viral DNA sequence and a short CRISPR RNA sequence there. And then two RNA molecules plus an enzyme, which is an endonuclease. And then at the third stage, the three components then assemble into what we call the ribonucleoprotein complex. And they assemble to form RNP complex consisting of the nuclease, CRISPR RNA, and the tracer RNA there. And these two components of tracer RNA and the CRISPR RNA combines to form what we call the guide RNA. And then these two components, or specifically this assembly of two major components, is what doing the cleavage, is what doing the gene targeting. You can think of it like a guided missile. You have a guide RNA with the viral gene sequence here that target the viral DNA, and then you have an enzyme that is akin to the missile. So basically, guide RNA is like the coordinate that you give to a missile to target which places that you want to detonate. But in this case, Cas9 nuclease targets DNA that has complementary sequences to the guide RNA and induces the cleavage or the double-stranded break. So let's look at it this way. First, the virus injects the viral DNA inside, and then the DNA is incorporated into the CRISPR loci, and the CRISPR system developed this guided CRISPR-Cas9 endonuclease system to target future incoming viral DNA. So in the future infections, whenever the bacteria is encountering the virus's DNA in the future infections, now you have this missile ready to attack. So the viral gene will be cleaved by the endonuclease and cause the double-stranded break, and then this DNA will be targeted for destruction. Okay, so that is just the basics. It might be confusing, but let's look at it in a broader prospect, in a broader view, so we can understand it in sequence. Okay, so the bacteriophage infects the bacteria, injects the DNA through the sheath there, and then the bacteria has evolved this system where it incorporates the foreign DNA, the viral DNA, inside the CRISPR loci, generate the guide RNA based targeting system, and then every time in the future infection, every time it encounters that same sequence, the viral sequence, the bacteria is now ready to cleave the double-stranded break, to cleave the DNA and cause the double-stranded break. And as you can see here, the how bacteriophage infects bacteria is by injecting the DNA. So the virus remains outside the cell, and the DNA is inserted into the cell. And if the RNA guided Cas9 enzyme cleave the DNA, there's nothing the virus can do. It just stays outside and be sad probably. So in response to that, the virus cannot do anything. So this is how bacteria has adapted the CRISPR system as an adaptive immune response toward viral infections. So now the table has been turned. Bacteriophage initially infect bacteria, and then the bacteria came back with an adaptive immune response that future viral infections won't be effective anymore. That is just the basics. And I hope that is not too much to understand. Okay. Now, how did all this system was discovered? Okay. So there are many scientists that have contributed to the overall discovery of the CRISPR system, starting from the 80s. But some of the prominent ones are Jennifer Doudna here, Emmanuel Charpentier, a French biochemist. Jennifer Doudna is an American biochemist. SIG, I think he's Lithuanian, and Zhang. And from this discovery, three of them, Doudna, Charpentier, and SIG, received the Kavli Prize in Nanoscience in 2018. So that's not long ago until people realized the potential of this system for genome editing tool, which comprises a gold medal and a share of $1 million USD. And of course, naturally, all this great achievement, you will be recognized. And only recently, in 2020, October 2020, in fact, which is only about 7 months ago, while we are still struggling with COVID infections, Jennifer Doudna and Emmanuel Charpentier managed to secure the Nobel Prize in Chemistry for the development of a method for genome editing. And these Nobel Prize winners, they are the first, it is the first time that two women jointly won the chemistry prize. It's never happened before. So of course it's a revolution for women in science, and it's something that we can celebrate. So congratulations. Now, how did they figure out how to use this system as a genome editing tool? I hope you have been explained about this, and you understand basically what happens. But in the end, all those that I explained before doesn't really matters. Why? You can ignore the virus, you can ignore the bacterial cell, and it turns out that if you synthesize the and purify the Cas9 nuclease and synthesize the gRNA recombinantly, form an assembly ribonucleoprotein assembly, and include any piece of DNA with the sequence complementary to the guide RNA, that nuclease can cleave that DNA just in a test tube. So it turns out that this system works in vitro. It is initially an evolved nature mechanism where the bacteria is developing a defense mechanism against the virus. But if you just take the guided missile out, you engineer the guide RNA sequence to target certain sequence of DNA, and you include that DNA in a test tube, that missile can target that sequence of DNA and cause double-stranded break, which is now interesting because then you can target any DNA, and we know every living organism has genomic DNA. So hypothetically, you can target any organism, given that you can access the genomic material. And it's also very versatile. All you need to target a sequence DNA, a sequence of DNA, is that particular sequence. This is probably in CRISPR-Cas9 system is about 20 bases. That has the NGG PAM sequence. Okay, you might think how that might be difficult, but it's actually really not. Take any piece of DNA. So this is a random DNA sequence of maybe about 500 bases, and I simply search for NGG. And then as it turns out, you can have those PAM motif anywhere in the sequence. Okay, so in these 500 bases, you can have about 10 or 12 targets. So those places can be targets for genome or genome editing or the double stranded break. And it's not only targeting one side of the bacteria of the DNA, because DNA is double stranded. It can target both strands. The one that goes that way, the 5' to 3', or that going that way, 5' to 3' that way. Okay, because in the end, the RNA is single-stranded and it targets any sequence that is complementary to it, and it induces double-stranded break. It doesn't matter if the sequence is complementary to the sense strand or the non-sense strands. And that is how versatile that system is. Next, the scientists have found out that you don't need to have two sequences of RNA, because they cleverly put a loop between these two RNA and link them together. And if you have a single-stranded RNA with the target DNA sequence, that can still bind to the Cas9 and form a ribonucleoprotein assembly or ribonucleoprotein complex, and still target DNA for double-stranded break. So now it's making it very simple. You just need two components with the sequence, with the guide sequence there, and it can target any DNA sequence. And then again, to be able to use this in a genome editing tool, as a genome editing tool, you don't need the CRISPR loci. Cas9 is a nuclease, it's an enzyme, and you can easily synthesize and produce enzyme by using the recombinant system in E. coli, because this Cas9 is a bacterial protein anyway. So if you use a recombinant system to produce and purify the Cas9 enzyme, you can do that without the need of the loci. And also, RNA is just a transcribed material from a DNA sequence, so you can do that recombinantly anyway. So in the end, all the stuff that I have explained before, although it is important for your understanding, all you need for a genome editing or genome targeting tool is simply the guide RNA that targets the complement sequence of your target DNA. This guide RNA will be fused together with the CRISPR and the tracer RNA, and this will form a binding with the Cas9 enzyme, and that is all you need. And what they found out is that in a eukaryotic cell, you can simply incorporate or insert the RNP assembly inside the cell, and then if there is a complementary sequence in the genomic DNA of the eukaryotic cell, you can simply target it. But this is not as simple. Eukaryotic cells, as we know, there are various different kinds of eukaryotic cells. You have plant cells, you have human cells, you have mammalian cells, and all with different structures. So it's not as simple as that. And CRISPR-Cas9, although the concept is very simple, the application is very wide and it's very species specific. And that is why I cannot explain to you all the different applications, but to explain only the basics, and then you explore it yourself. And of course, in a eukaryotic cells, you have a nuclear envelope there. So there are multiple barriers that you need to overcome to be able to deliver the Cas9 RNP to the genomic DNA. And that is one of the simple limitations of the CRISPR system. Now, we haven't really gone to the genome editing bit. All we have gone through so far is that we are able to utilize the CRISPR defense mechanism in the bacterial cell and use that as an offense mechanism, where we inject or we insert the RNP assembly into the eukaryotic cell and cause a double-stranded break in the genomic DNA. And as the scientists were studying this, of course, now this is now an offense mechanism. So if the DNA in the eukaryotic cell is damaged by double-stranded break or cleavage, of course, the eukaryotic cells have to respond to it, because we cannot have a break in our DNA and which could destroy the genomic integrity of our genome. Now, we don't want our genes to be affected because, you know, we are eukaryotic cells with many different metabolism and mechanisms. So a damaged gene could lead to a really widespread damage. So in comparison to the virus, the defense mechanism of bacteria to the bacteriophage before, as I said earlier, the virus is only at the outside. And as the bacteria is destroying the viral DNA inside the cell, the virus cannot do anything. But when you use this system the other way around, where you use the RNP complex to target the DNA, of course, the genomic DNA is inside the cell, and the cell has to respond to it to maintain the genomic integrity. And it turns out, if you do that and cause the cleavage in eukaryotic cells, the eukaryotic cells respond in one of these two pathways. Okay, I wouldn't explain these pathways in detail, but it's either going to repair the cleavage by using the non-homologous end joining mechanism, or simply NHEJ, or the homology directed repair. I'll explain a bit about the difference between the two. So as the Cas9 is cleaving the genomic DNA, now there's a double-stranded break there. And then it is the responsibility of the cell to fix the break to maintain the genomic integrity. And then in non-homologous end joining, what it does is just it uses a random pieces of sequence of DNA and fix the break by introducing several DNA sequences inside, so that it is now intact. But doing this often result in insertion and deletion. So insertion and deletion is where you have a gene that is cleaved, and then in the non-homologous end joining repair mechanism, you will have either insertion of other sequences into the gene, or a deletion where it is shortened a bit. And of course, if that is a gene, and then you have an insertion or deletion, it is a mutation. And depending on the nature of the insertion and deletion, you can obtain different results. For example, if there is a 1 or 2 bases inserted into the break, then that has destroyed the frame of the codons, and then of course that gene will from that point onwards would be affected. But if it inserts 3 bases, at least you have an insertion with a new amino acid, and that at least doesn't change the frame. But of course, there's an introduced mutation there, which could affect the final product later. So this non-homologous end joining is a slightly random repair mechanism. It can happen in many different ways, and you can have many different insertion or deletion depending on what you want in your experiment. So if you use this CRISPR-Cas9 to target your eukaryotic cells, what you get in the end is either insertion or deletion in this if it is repairing through this pathway. And then this pathway is suited for scientists that are trying to do knockouts or gene knockouts, because simply if you insert random sequences, then the frame would be affected and then the gene would be deactivated and you don't have the right protein. So that is a gene knockout product. But the other pathway, this is the one that is really the attention, the thing that people are paying attention to right now. Why? Because if you provide a donor DNA, or what we call a repair template, once the Cas9 introduced the double-stranded break there, and then the repair template normally have a sequence that is homology to the region that is cleaved, and then you can insert anything in between in the repair template. And what happens is the cells will carry out this process called homology directed repair, and this would integrate the donor DNA into the sequence. So now this is more programmable, where you can introduce any gene sequences, you can introduce any site-directed mutagenesis, site-directed mutations. You can introduce it is a really controlled process where you can program what you need and engineer what you need to put inside the genome. So this is what this is how CRISPR-Cas9 system has been adapted for genome editing tool. So has it been successful? Yes. So CRISPR-Cas9 system has shown to be successful in human cells, in zebrafish models, in C. elegans, worms, your friendly neighborhood fruit flies, plants, and also primates. So as you can see, as I talked about earlier, as long as you have the RNP complex and you can have that RNP assembly access the genome, you can hypothetically target any organism. So let's now think about the vast potential of this technique. If you can target virtually any organism, food security. If you use CRISPR-Cas9 for plants, you can make plants withstand drought or disease, and then you can solve problems in related to food security. Climate change. If you can modify forests and you can have a forest that is more environmentally friendly or more sustainable, then that could ultimately help climate change. Bioenergy. If you can engineer a bacteria that can harvest sunlight and produce a sustainable energy, imagine how that would help solve the energy problems that we have right now. Healthcare, for example, this is one of the main application of CRISPR-Cas9 at the moment, where you can edit mammalian cells, edit cancer cells, and you can use this CRISPR system for medicines. In fact, there have been CRISPR system used in targeting cancer cells, and there's a hope of using this system in treating cancer. So I guess that would be all for now. I hope that is not too intimidating. It's basically a basic CRISPR-Cas9 introduction. It's just a window so that you understand how it works, and now it is up to you to explore more, and hopefully the talk by our next panel, Dr. Asyraf, would kind of put things into place of how you utilize that CRISPR-Cas9 system in plant. So I would like to thank everyone that has shaped me into who I am today. Professor David Rice was my PhD supervisor in University of Sheffield, and all these Sheffield folks, Dr. Patrick Baker, John Rafferty, my colleagues Sam, Alicia, and this lab staff, Fiona Rogers, and Sveta. And I'm also very highly indebted to my ex-colleague in Institute for Medical Research, IMR, where I have spent quite large amount of time studying CRISPR-Cas9 system. And these are the list of names. Dr. Aziz there was my supervisor. Dr. Shamila is the head of unit. Dr. Ellen Nurfer, a big shout out to her for helping me understanding CRISPR-Cas9 just through emails. So I kept sending thousands of emails to her, and she kept replying back regardless of how busy she was. So I learned a lot about CRISPR-Cas9 from her, and she is a collaborator, a UK collaborator to Dr. Azian. So I know it is not so advanced, but I'm sure some of you who are only beginning to understand CRISPR-Cas9 can see the potential of using it for your research. And I hope my short talk there could pique your interest and get involved in doing research by using CRISPR-Cas9 to help solve global problems. And I really recommend you to watch the Nobel Prize lecture by Jennifer Doudna on YouTube, because that in her talk she explains how the CRISPR-Cas9 was used in a really in layman terms, and you can really be inspired from her talk, and that's what all we scientists need, a little bit of inspiration. So thank you for listening, and that is, I guess, all the time I have for today, and have a nice day. Back to you, Dr. Mima.
So thank you, Dr. Adli, for your great sharing on the insights of CRISPR-Cas9. So I also learned a lot today. So starting from the bacteriophage, infects the bacterial host cells, and then develop the adaptive immune response to encounter next viral infection using this CRISPR-Cas system. And then it's such a brilliant ideas. And then we human, we adapt this system in our research using RNA complex to do the genome editing, right? So just to remind the participants here, the question and answer section will be after our second section. So you can drop your questions now using the Slido link given in the chat box, also in the Facebook comments. So ladies and gentlemen, I am pretty sure now you would like to know how can we apply CRISPR-Cas9 knowledge in plant research. So, but before that, let me introduce the biography of our second speaker. So Dr. Muhammad Asyraf Hatta. So he's a senior lecturer at the Faculty of Agriculture, University Putra Malaysia, and has worked for 3.5 years in the field of plant molecular biology. So he earned a Bachelor of Science in Biotechnology at International Islamic University of Malaysia, and a Master of Science in Plant Biotechnology at Wageningen University and Research Centre, the Netherlands, and a PhD in Biological Sciences at University of East Anglia, John Innes Centre, Norwich, United Kingdom. So he teaches agricultural biotechnology and agricultural biochemistry courses for undergraduate students. So during his PhD, he attempted use CRISPR-Cas9 for in vivo gene stacking in cereal crop, and is currently applying the technology for the purpose of improving important agronomic traits in crops. So next, I would like to invite Dr. Muhammad Asyraf to give his talk on how to apply CRISPR-Cas9 techniques in plant research. So Doctor, now the floor is yours.
All right, thank you very much to our moderator, Dr. Nurhamimah, for the nice introduction. Are you able to actually hear my voice here? Can you see my slides?
I can see your voice, but I'm not able to see your slide.
How about now?
Okay. Yep. That's good now.
Perfect, right. Okay. So Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to our honorable guests and all members of the audience. I'm very pleased to be able to join this webinar today, and we have, I would like to talking about CRISPR-Cas9. So we have already heard about this technology in the last presentation by Dr. Amirul about the origin and the mechanism of this system and how the scientists can manipulate the system to engineer even, for example, better crops. So, but now I would like to look how this can impact the work in our important crops. And my talk today, I will firstly explain about the various application of CRISPR-Cas9 in crops, and hopefully I can show you this system, this technology is a valuable tool to create genetic diversity or genetic variation for crop improvement, and how this technology can fit into the breeding techniques that we currently have and applied by the breeders, and then different genetic manipulations, what we can do using CRISPR-Cas9, and then giving you some examples, but focusing on the transgene-free edited crops, and I'll give you the reason why later in the next few slides. And then the second part of my talk, I will go into more practical part, how to plan your experiment, but focusing on generating targeted gene knockout in crops, and we're going to go through the steps from selecting your target sequence, designing guide RNA, and until the screening for the possible mutation. But I will explain these steps briefly, just to give you some idea. Right. So plant breeders have done incredibly, a fantastic job for the last hundreds of years. For instance, as you can see in the slide, they bred a wide range of rice varieties that we have today, from wild species that you can see on the left. Okay? And this is quite an achievement, and this achievement is possible because the breeders have an access to variation. Okay? And so they have the access to the very small changes in the DNA that enable them to select things that, for example, like pigment color or grain size or grain shape in the case of rice. Okay? And so the way this has been done, usually by finding the variation that needed from closely related species. So in the classical breeding, so the breeders make a crosses between wild species and an elite cultivar to transfer their targeted gene or their desired trait, shown by this orange star here. However, during the process, they're also bringing in blocks of DNA from the wild species that might be, something that they don't want. So they still have a lot of work to do to remove all this background from the traditional materials. Otherwise, this might, affect the performance of the crops, and that's known as linkage tract. And then the breeders, if they, if they can't find the variation that they needed, so they look for other ways to induce the variation. And in the past, for example, one method is the mutation breeding, in which the breeders use it back in 1940s. The seed treated with either chemicals or radiation to deliberately make changes in the DNA. And some changes that they can see by chance might hit their target gene and create a variation, but some of them think that they don't want. So again, the breeders have a lot of work to do to remove all these background mutations and select the one that express the desired trait. And then jumping forward, of course, we have genetic modification, and in this technology, the breeders and plant scientists are able to insert their gene of interest into their plant species they are working with. However, this end up in random location, so we are not controlling where the genes goes in, and it's not usually a problem. You just need to do a little bit more work to get what you want. So you will get probably some other random mutation along the way, the tissue culture process. And now we got a new technology, and this is one that everyone get excited about, and which is the CRISPR-Cas9, which is one of the tools in genome editing. And instead of having to use the process that I have mentioned before, we now have the option of going directly into the plant that we're working with, finding the genes, and then making the changes directly at the gene, which is shown here in the green color. And apart from making site-directed mutagenesis, there are also other applications. Dr. Amirul have talked about the gene knockout, and also another one, gene knock-in, where you can introduce a fragment of DNA into the genome by providing donor DNA that have homologous sequence the locus that you want to target. And then over the years, since this technology has been adopted by many laboratories around the globe from 2011, then they have there are other studies that have expanded this the application of CRISPR-Cas9 not only for gene knockout and gene knock-in, but now they can also use CRISPR to, for example, overexpress your target gene or silence the target gene, and also make base editing. So in this application, the Cas9 mutated at one of its domain, so it will you're not able to induce or create double-stranded break, but instead this known as deactivated Cas9, dCas9, will can be used to direct the either activator or repressor or deaminase to the targeted location in the genome to do the downstream process. So in crops, there are many examples and studies that have been shown to that they successfully use this technology to develop or target genes for example, higher yield, better quality, enhanced nutritional value, and also disease resistance. So these are some few examples from the long list of the previous studies that shown the successful of this technology in plants. But as I mentioned before, I would like to focus in this talk to this transgene-free edited crops. Okay, because at the moment there's a huge amount of discussion going on whether to regulate these edited crops using CRISPR-Cas9 or any other genome editing tools as genetic modification or GM or non-GM. Okay, and one of the arguments for countries that still regard this as GM is because there is a presence of transgene genome of the plant for doing the editing activities. So, and then the plant scientists have developed few methods to generate transgene-free edited crops. And the first method is the most common one, in which they deliver the CRISPR-Cas9 component into the plant cells, and then produce the T0 generation, in which some of the plant will carry the mutation and also the transgene, and some of the plant do not carry the mutation, but the transgene. And then in the second and the next generation, the T1 generation, so all these progenies are segregated based on Mendelian segregation. So some of them will be homozygous, some of them will be hemizygous, and some of the plant will be null. So in this case, in the case of null segregants, then when we do the screening, we actually will be able to identify the plants that has been mutated but do not carry transgene in the genome. And this is possible because when we deliver the CRISPR-Cas9 component, the transgene is inserted in the different location as the one that the target site. So in that case, in the next generation, this can be segregated away. And then for the second method, it's kind of similar with the first one, but in this method, suicide genes, Barnase and CMS gene are introduced along with the CRISPR-Cas9 component into the plant cells, and in the T0 generation, there will be some plants that have mutation and some don't. And then in the T1 generation, those who have these suicide genes won't be survived and then died, and you will only recover plants that have the mutation, which is the null segregant. And then in the third method, this is a transient expression of CRISPR-Cas9 component, either in DNA or RNA component. And transient express means there is no stable integration of the CRISPR-Cas9 component into the genome, but the component is only transiently expressed in the plant cell. And then using this method, they transfect the protoplast, and some of the plant species have the ability to regenerate from protoplast, and in T0 generation, they can identify null segregant, so don't have to wait for the next generation. And then in the fourth method, similar to third one, but instead of centrally expressed DNA or RNA, now in this one is a DNA free, means that the CRISPR-Cas9 component will be transiently expressed in the form of ribonucleoproteins, RNP. So in this case, the CRISPR-Cas9 component will be transiently expressed, induce the mutation, and then will not integrate it into the genome. And then you will get non-segregant that have only that carry only the mutation. So one of the examples of a previous study for the first method that isolate the non-segregant by Mendelian segregation is the generation of tomato variety that is resistant to powdery mildew fungal pathogen, and they took less than 10 months to generate this transgene-free tomato plant. So they targeted the susceptibility gene in the plant, tomato plant, that contributes or promotes the growth of the pathogen by knocking out this gene MLO1. So in the edited crops, they can see the plant confers enhanced resistance to powdery mildew. And this is a typical analysis done in the CRISPR work, in which you will align the sequence from the wild type and then with the putative mutants, and most often the double strand break will occur 3 to 4 base pair upstream the PAM motif. So Dr. Amirul have explained about the PAM sequence here. So normally the double stranded break will occur 3 to 4 base pair upstream the PAM motif, and generate and the repair is most of the time imperfect, and this can induce mutation in that location. So in this is one of the screening methods. So I will go into detail about this when it comes to screening the mutants. And then the second example, using the method, transiently express the CRISPR-Cas9 component in the form of DNA or RNA. And this work has been done in wheat, in which they coated the DNA RNA of CRISPR-Cas9 component and bombarded into callus and regenerate into plants and screen the possible or putative mutants. And they were able to identify one of the plants to carry the mutation at, all at 3 different genomes. So wheat is a hexaploid plant, and it has 3 genomes, A, B, and D. And in the past, it's really difficult to simultaneously induce mutation at all 3 genomes at the same time. So using this method, they were able to identify progenies of plants in the first generation, the T0 generation, that carry the mutation at all 3 genomes. And then in the next method, transiently express, instead of transiently express the DNA or RNA, there are many other examples or previous study that use purified Cas9 protein and the transcript of guide RNA, pre-assembling this as a complex, and then transiently express this in the protoplast of different crops like tobacco, lettuce, rice, and also the modest species Arabidopsis thaliana. And they shown that this can be done, and they achieve mutagenesis frequency of up to 46%. And this is in this figure here, they show one of the plants. They were able to regenerate into a whole plant from the protoplast and actually carry the mutation right in the T0 generation. So the RNP complex might become a prominent strategy for plant genome editing, because this CRISPR-Cas9 component is not integrated into genome, and we can avoid the transient integration or any foreign DNA. And then this also have shown to reduce off-target effects. So I explain about the off-target effects in the next few slides. But the point that I want to make here is that going forward, this method using RNP complex might be the way to produce the edited plants, which might, deregulated or exempted from the GM regulation. And other studies include the one in wheat and also in maize. But important point to make in this for this method is might not be possible, or might be a significant bottleneck for crop species that are not easily regenerated from protoplasts. So the regeneration for protoplasts have not yet been established for quite a lot of plant species. Right, so now I would like to move to the second part of this talk. Just briefly explain to you the step to how to plan your experiment for targeted gene knockout. Okay, the first step is the selecting the target sequence. So you might want to check whether your target gene is present or not in the cultivar of interest. And then when possible, you also might want to sequence the region to modify prior to designing the guide RNA in case there's a sequence variation, because if there's any, then this might result in reduced cleavage by the Cas9. So the checking of the presence of target gene normally can easily be done by designing primers and then amplify the target gene in the cultivar of interest. And you can also do this Sanger sequencing or even next gen sequencing. Now the cost is not as dropped significantly, so you can sequence the region that you want to target. And then once you have your target sequence, now designing the guide RNA. So if you want to knock out the target gene, whether it's only one gene, are you targeting only one gene or several genes or gene family? Okay, so a lot of studies have shown that multiple gene knockout can be achieved quite efficiently. So you can also do this in case if you have several targets. And then how many guide RNA for each target? So usually 2 or 3 guide RNA will be designed for each target, so that in case one of the guide RNA didn't work, so you have the other 2 as a backup. And then for gene knockout, we normally design the guide RNA that target the 5' of the gene, right? Probably right before the ATG or in the first exon, so that you can hopefully get early stop codon in your target gene. And the target sequence is consists of 20 nucleotides. And this 20 nucleotide sequence followed by a 3' PAM sequence. And the PAM sequence that from the common Staphylococcus aureus Cas9, as explained by Dr. Amirul, and the sgRNA is expressed using an RNA polymerase III dependent promoter, for example, U3 or U6 promoter. And these promoter are used for driving this single guide RNA because of the precise transcription start. So for example, U6 promoter, the transcription start with a G, and for U3 promoter, the transcription start with A. So you can select a single guide RNA with 5' G, and then followed by the 20 nucleotide bases. Or if there's if the target sequence doesn't start with G, then you can add a G, because it's not necessarily that you have the same sequence as the genomic DNA. Okay. Then the sequence, the 6 to 8 nucleotide sequence, immediately 5' of the PAM are called seed region. So you have the PAM sequence here, NGG, and 6 to 8 sequence upstream the PAM sequence is known as seed region. And this seed region should be 100% identical to target sequence to be so that the Cas9 will be able to bind and then cause double strand break. And then the remaining 12 base pair will be, if there is any multiple mismatches, this can be tolerated. Okay, and this is really important when you design the guide RNA to make sure that you don't have any similarities in this seed region with any other location in the genome. Otherwise, you will create off-targets effects. So that's if you can use the criteria if you want to design your single guide RNA manually. But nowadays we have many web tools available that you can use for plants, and this is the list of the available web tools. And the most common one is CRISPR-P, Finder, CRISPR plant version 2, and in my lab, my group, we are using benchling as a tool because this is quite convenient. So I'll show you the examples of this, the output from this benchling in the next slide. Just one point to, one thing to point out here, if you design one guide RNA, you might want to find a selection site, a restriction site enzyme where the mutation, if there is any in that particular location, will result in loss of the restriction site, and this can help you greatly when you do the screening that and I'll explain later. And if you design 2 or more guide RNAs and you hoping that you will create small deletion between them, so this also will be something that you might want to consider. So please do visit to each of these web tools, because sometimes probably the crop that you are working with, the genome is not available in this one of these tools. So it depends, really depends on the species that you are working with, and if there's not any web tools available, then you can still design manually. So as I mentioned before, these are examples of the output when you design the single guide RNA using benchling. So it's very simple. You just input the sequence, the target sequence in the query, and then this list will come up when you select the region in the target sequence in which you want this program to identify any single guide RNA. And this tool also will give you a score on target and also off target, so that's really convenient. So there's a certain, specific algorithm by this tool that they calculate the on target and also off target score, but the higher the better for both. So in this case, for example, for this one that I selected here, it has 67 score for on target and then nearly 100 for off target score. So this is quite a good one. And then if you look here, this is a negative strand. So DNA has 2 strands. So this is for the complementary strand. And in the sequence here, when you click the selected guide RNA, so it will appear in the sequence, the location of the target sequence. And then if you can zoom in here, so this is negative strand. So the PAM motif here, the AGG will be on the complementary strand. And this is a big PAM sequence. And as I mentioned before, the double strand break will normally occur 3 to 4 base pair upstream the PAM. So in this case, the double strand break and possible mutation will occur between C and G. Okay, so in this case, it's really convenient to have recognition site of an enzyme. And this tool, one of the features that really helpful is that it can suggest any recognition site at the target sequence. So in this case, for example, enzyme BBVC1 will have this recognition site, and is cover the possible double-stranded break location. So in this case, having this by having this recognition site at this point, so if there is any mutation occur here, so you can when you digest the genomic DNA with the enzyme, then the wild type will be digested, but the one that have been mutated here, that have the possible mutation, will disrupt the recognition site so that it won't be digested. So then when you run on gel, you can differentiate based on the amplicon size. Okay, so I'll go into that more detail when we come to the screening of the mutants. So now I'd like to move to the off-target. So why off-target is so important, and I'll give you some examples of off-target in plants and how to predict off-target. So off-target is occur when the Cas9 cut the location in the genome that have sufficient homology to the target sequence. Okay? So you have 20 nucleotide target sequence followed by the pan motif. So if there is any mismatches like SNPs or small insertion or deletion, about 1 to 2 nucleotides, so then the Cas9 will wrongly target this location instead of your actual target site and cause double strand break at the undesired location and might cause mutation in other genes rather than your target gene. So this is this off target here, because of the partial mismatches, might cause might affect the expression of the non-targeted genes and produce undesired phenotypic expression. So this is something that you want to avoid when you design the guide RNA. However, in general, off-target is rare in plant, and in this study in rice, they show that most mutation in edited plants, so they screen a lot of edited plants, and they found out that most of the mutation are created by tissue culture process rather than off-target effects of Cas9. Okay? And they also suggest that Cas9 nuclease is very specific in generating targeted genome editing, and you can avoid these off-target effects if you design the guide RNA as specific as possible. So although it's rare, but there are some examples, very rare examples in plants where you get off-target effects. In this study, they targeted 2 genes, each one from barley and also from Brassica oleracea. And in barley, this is the actual, this is the first target. This is the first guide RNA for the target gene in barley, and this is the original target sequence. And the scan through the genome of the barley, of the species cultivar that they are working with, and they identified 2 possible off-target sites that have almost similar sequence with the original one, but only one position different, like for example in this one at nucleotide number 9 from the PAM sequence. And the second one also a single base pair difference. And from these 2 off-target sites, they didn't get any mutants from one of them, but in one of the off-target site, 4% of the mutant has been produced. And then in the second single guide RNA, they identified another 2 off-target sites, but in this case, there are no mutants generated from this experiment. So if you can look here, as I said, it's really crucial to have 100% identical sequence at the SIG region, which is 6 to 8 base pair, in case if you want to avoid any off-target. And in this case, indeed, there's no in the SIG region. And then for the Brassica oleracea, again, they have 2 targets here, and then each of them have identified 1 off-target site. In one of them, there's no mutant having produced, but in the other one, it's quite high, which is 36% of the mutants have been produced from this off-target site. And there's another examples in rice in which they identified 3 off-target sites. 2 of them didn't produce any mutants, but 1 of them produced a mutant. And in this one, there are 3 mismatches, 3 different base pair or nucleotides with the original target sequence. So although the off-target is rare in plant, but it can happen, and it's really important to design your single guide RNA as specific as possible. How to predict this off-target? Similar to single guide RNA design, there are also programs or web tools available, and each program have different strength. And as I said earlier, some of the program might have your the genomes of your crop that you are working with. Some of them not, and some of them might allow use of custom genome take requests and other strengths here. So the examples of web tools that allow you to predict off target, I put it here in this table, and one that I use currently is the Benchling, and there are also other ones that might be useful for you to use. Right. So after you design your single guide RNA for your target sequence, then you can start to design the oligos or primers so that you can amplify and then clone into your desired vector. And you can clone this into the plasmid containing the guide RNA scaffold using several DNA assembly methods like standard precision ligation, golden gate, and so Gibson assembly. And all these plasmids are available from Adli or from the commercial companies. And this is how the binary vector, the final vector, look like. So you have the selectable marker gene cassette, your Cas9 cassette, and then followed by the single guide RNA, either 1, 2, or can be more than that. So you can have multiple single guide RNA. And then the DNA assembly method really depend on your resources in the lab. The one that I'm using is golden gate cloning. So this is a very rapid and then convenient method to assemble different parts. Unfortunately, I don't have time to explain this in more detail, but you can read the paper in this that published in 2014 that described and explained about how this golden gate cloning work. And alternatively, you could also watch a video in the YouTube made by my former co-supervisor when she worked in the Sensible Lab in laboratory in Norwich. And this is a series of videos where she's showing you how you to use this golden gate cloning to assemble different parts of DNA. So for the single guide RNA vector, after you amplify this 20 nucleotide target sequence, then you can clone it into with the promoter. But you can amplify this using a template that have the single guide RNA scaffold. So this is one important part of the single guide RNA, and then clone it in with a promoter in a level 1 acceptor. And the promoter that you can use either promoter U6 isolated from wheat for monocot species if you are working with monocot species, or you can use U626 that is isolated from Arabidopsis thaliana for dicot species. So basically you will carry out level 1 reaction using golden gate and to assemble the single guide RNA with your promoter. And then for the Cas9 and also selectable marker gene, these are some options that you can use for promoter. 35S, double 35S, maize ubiquitin promoter, nostamine promoter, and so on and so forth. And then for the coding sequence, we are using Cas9 from Streptococcus pyogenes. You have a human codon optimized and also plant codon optimized. So there are some available options for you to use depending on the species that you're working with. And then for the selectable marker gene in plant, you can use either NPT2, hygromycin, HPT2, or bar genes. And then the terminator selection here for Cas9 and selectable marker gene, 35S, nose terminator, OCS, and also G7. So one point here, this list or option is not limited only to this one. You can also find other promoter, terminator from the ad gene. So please have a look in the this non-profit depository plasmid organization. So once you have assembled your construct or vector, you can introduce or deliver this into plant cells, either as plasmid DNA or RNA or RNP. So, and there are several methods to directly deliver this CRISPR-Cas9 component, either by ballistic bombardment, nanoparticles, PEG, chemicals PEG, and also plant virus. And the most common one is the agrobacterium. So again, this really depends on your species that you're, the plant that you are working with. Some of them, can easily be transformed using agrobacterium. But in the case that you have a, it's not susceptible to agrobacterium, then probably ballistic bombardment will be the way to go. And then once you have produced the regenerated plants, expressing the CRISPR-Cas9 component, and now you want to screen the plants that you have regenerated to find which one carry the mutation. So there are several methods available, developed in the past, like PCR band shift assay, restriction site loss assay, survey assay using cell 1 and T7 endonuclease, or high resolution fragment analysis, HRFA. And of course, once you have identified putative mutants, then you can double confirm by sequencing and looking at the phenotype. But in this talk, I will talk in detail for the first two methods, because this is the most common used by the plant scientists. So in the first method, PCR band shift assay. So this is the case if you design two single guide RNA. You have target 1 and target 2, and then you intend to induce a small deletion in flanking these two targets. Okay, and you can screen the regenerated plants by extracting the genomic DNA, and then use the DNA as a template to amplify using forward and reverse primer that flanking these two targets location. And this actually can create, if you design the two guide RNA, there's a study showed that the deletion can be detected up to 120 kilobase pairs. So it's quite a huge size, and even 250 kilobase pair. But one thing to bear in mind that the larger the deletions, the lower the mutation efficiency that you get. Okay, and the most efficient one will be less than 100 base pair deletion size. Okay, so from the genomic DNA, you carry out the PCR using the primers, and for the wild type, when you run on the gel this PCR product, for the wild type, you will see amplicon at the upper band here, and then if you if there is absence of wild type band here, but instead there is another amplicon with a smaller size, then this indicates that you might get the homozygous deletion, meaning that the deletion will occur at both alleles if it's a biallelic. So there's because of the small deletion here, now the amplified fragment will be smaller in size, hence you see lower band here. In the case of a mix between amplicon similar to wild type and also amplicon at the lower side, then this might suggest that the plant that you screen might have or carry heterozygous deletion, meaning that only one of the allele has been deleted. Okay, so this is one of the method to identify your putative mutants. And then
Sorry, Doctor. Sorry for interrupting. You have another 5 minutes to finish your slides. Is that all right?
Yeah, sure. Yeah, I'm almost finished. Okay, and then for the undigested, in the case of if you get amplicon that similar to the wild type, but there's a thin band at the lower side, so this might indicate that the deletion amplicon is low or absent. Okay, so in this case, you can, if you have a restriction enzyme endonuclease site in the deleted region, so this will come handy for you to differentiate between wild type and the mutants. So if you have the restriction enzyme site here, once there's a deletion occurred for mutation or editing events, then during the amplification for wild type, you won't get any amplicon amplified because now this has been cut or digest into two parts, and then the PCR will be able to amplify. And then for the one that has been mutated and deleted, there's a restriction enzyme site which is disrupted, and you can still amplify and then digest, and you will see a lower band here. So that's if you have two single guide RNA. So in the case of only one guide RNA, as I mentioned before, the double-strand breaks are mostly occur 3 to 4 base pair before the PAM. So if you have a PAM here, so this might occur around here. And when you design the single guide RNA target, you might also want to look for restriction enzyme site that have the recognition site at this target locus. So if any induced mutation or imperfect repair by the cell, then this restriction enzyme site will be disrupted and you won't be able to digest this amplicon. So for undigested amplicon, you can't really differentiate between wild type and mutation because if there's any indels in there, it's a very small changes, small difference. So by digesting your PCR amplicon with this restriction enzyme, so you will get 2 bands for wild type. And if there's a mutation there, in addition to the 2 amplicon size, there's another extra band upper side that correspond to the restriction enzyme, restriction endonuclease resistant band. Means that this band is produced as a result of the non-digested due to the disruption of the restriction enzyme due to the mutation. So this is another way to distinguish between wild type and mutation before you sequence the amplicon. So as a summary, we've gone through this part 9 here for targeted mutagenesis in plants by first selecting your target sequence, and then design your single guide RNA. You can design manually or using web tools or combination of both, and then assemble using different assembly method like golden gate cloning or Gibson assembly, and then deliver this construct into plant cells, and then obtain the regenerated plant, and then start to screen for mutants. And we have covered 2 of the most commonly used methods to screen for the mutants. So I hope from this talk, I've convinced you that, and also from Dr. Amirul's talk, CRISPR-Cas9 is the fastest, cheapest, and most precise way of editing genes. And this tool can be used as a valuable tool to generate better crop with enhanced resistance, for example, to disease, improved yield, and also quality. And I've shown you some examples of previous study that successfully used for targeted mutagenesis. And apart from gene knockout, this CRISPR can be used for other application like single base editing, overexpression, gene silencing, also gene tracking. Okay. With that, thank you very much for listening, and I'm happy to take any question.
Thank you, Dr. Asyraf, for the very insightful presentations, like how you can apply CRISPR-Cas9 to target genes to improve crops for higher yield and better quality. And also from Dr. Asyraf's presentation, also you guide us like step by step from selecting genomic target, how to design the guide RNA intermediate vector, softwares to use for the designation, tagging the off targets, the available Cas9 and selectable marker gene cassettes, and also how to deliver the vector components into plants, and also checking the mutations. That's a very, very detailed and very complete presentation. Thank you so much, Dr. Asyraf. So now, ladies and gentlemen, I open the floor now for the questions to our panels. So for the Google, for those in the Google Meet, you can turn on your microphone to our panels, or you can use the chat box. For watching us in live streaming, in BioMECs Facebook page, you can type your questions using the Slido link given in the comment, or you can scan the QR code as displayed on the screen. So there are a few questions come up in Slido link. So Dr. Amirul Adli, are you there?
Yes, I am.
Oh, okay. So the first question, let me check. The first one, do you have any information on, probably this question is for Dr. Asyraf. Do you have any information on how CRISPR edited crops will be regulated in Malaysia? Will it be excluded from GMOs regulation like in the US or Canada?
Yes, that's a very good question. The multi-dollar question for Malaysia. At the moment, as far as I'm aware, there's no clear guideline how we regulate these CRISPR-Cas9 edited crops. But in some countries, they have made a decision on that. For example, in the US, they really look at how the method of producing this edited crop, either is any foreign genes that's been introduced, and they're quite open on that, similar to GM crops. And for other parts of the country, like in the EU, so the most recent one from the EU ruling court, they regard this as GM. So there are still a lot of debates going on at the moment around the world whether to regulate this as GM or not.
Okay, thank you. So next question from Nurul Ashikin Muhammad Zim. And how do you think the acceptance of the public if genome edited crops to be commercialized? What do you think, Dr. Asyraf?
Yes, as I mentioned during the talk, there is a huge potential of commercializing this edited crop if we can produce and generate the edited crop without introducing any foreign gene, and also undistinguishable with the one that we generate using traditional breeding methods like mutation breeding. So if you can make a mutation or induce a mutation, a similar one as the previous method, but this time is with greater flexibility and precision. So I don't see any arguments that can, stop us from applying this to produce a better crop. In fact, for GM or gene edited crops, if it's regarded as GM as well, they will be, they will go through a lot of regulation process, tests to really make sure that this is, safe for human consumption.
Okay. All right. Next question from Azwan. He has two questions. So maybe the first one, Dr. Adli, it's meant for Dr. Adli. The second, maybe either one of you can answer. So the first one, any way to selectively induce NHEJ or HR? And the second question, any regulation requirement to conduct CRISPR experiments in Malaysia?
Okay, I'll just answer both questions, I guess.
Okay.
Yes, I showed in my slides earlier, there are two pathways for the repair mechanism in eukaryotic systems. You either have the NHEJ or the homology directed repair, HDR. It's not just the way of selecting it, but this pathway is also species dependent. Some eukaryotic cells have a poor HDR mechanism or the other. So, for example, the eukaryotic organisms I was working with at Institute for Medical Research, malaria cell is a parasite. Malaria has a really poor NHEJ pathway mechanism. So if you introduce a double-strand break on a targeted gene, it fails. It has the difficulties to use NHEJ to fix or to repair the double-stranded break. So if you induce the double-stranded break, then most likely you have damaged the gene, and then of course downstream effect would follow. Only if you produce or you include together a repair template, which is the donor template DNA that has homology to the sequence surrounding the double-stranded break, then malarial cells would selectively prefer the HDR pathway and fix that double-stranded break using that pathway. So to answer that question, it is very species dependent, and some species can have both pathways, but if you include a donor template, they would go towards the HDR pathway and selectively use the donor template and carry out the HDR pathway instead of the NHEJ. So it is basically a try and error method, and then figure out what works for your cells, your eukaryotic cells. And again, you can only know if you try that system. And I mean, malaria have had some people studying it, and then we have established some data on which and how to select the pathway. But again, it's species dependent. So if it is not published in articles, if scientists have never done it before, then it's always a try and error situation. The second question, again, regulations for conducting CRISPR, again, it's species dependent. If you are studying malarial cells for medicine, then and of course, depending on your aim of your project, what are you trying to mutate? Are you trying to introduce a mutation that would release a drug-resistant strain into the public? Of course, that wouldn't be allowed. So there is no, I don't think there is a general regulation for using CRISPR-Cas9 technique, but there will be some ethical considerations considering the species you are working with, and that you have to refer to your ethics committee.
How about CRISPR implants, Dr. Asyraf? Is there any regulations for that?
Yes. So at the moment in Malaysia, if you are using agrobacterium as a method to deliver your CRISPR-Cas9 component, then you have to you need to have approval from the council for the genetic modification work. So that's something that you need to bear in mind. So you need to go through any relevant trainings, so that we know how to handle these materials, how to discard them when you have done your work in the lab.
Okay. So we go for the next one. Aziz from UIA Kuantan. For Dr. Asyraf, you state in your off-target slide that most mutations are created by tissue culture process. Can you elaborate on that and how to prevent it?
Yes. I think the mutation is
Random.
it can occur naturally. Okay, and so since that's one of the arguments as well, if you want to regulate the CRISPR-Cas9 edited crop as a GM, because there are some studies showing that the mutation, even the horizontal gene transfer can occur between one plant to another, from one species to another. And in the case of tissue culture, this is also can happen when you do tissue culture work. There will be some mutation occur during the process because they are now regenerating from any part of the plant, and during the process there might be some mutation.
All right. Okay, the next one from Nurul Ashiki Muhammad Zim. So, what are the differences between Cas9 and D-Cas9? I think D is that Cas9, isn't it?
Yes. Yeah.
Okay. So, what is the difference?
I don't know if Dr. Amirul would like to respond on that.
So D-Cas9 is a mutated Cas9 enzyme. It's a mutated endonuclease in which the cleaving capacity is removed. So the Cas9 nuclease can no longer, is mutated so that it can no longer introduce the double-stranded break. And then you will wonder, why do this? Why, if you can't do the double-stranded break, then you cannot induce the indels or the knock-in or knockout. That Cas9 enzymes are used for other applications such as reporter assay. For example, if you use the guide RNA and the nuclease only to target the DNA so that the Cas9 binds to it without cleaving the DNA, and then if you have a reporter gene attached to the Cas9, for example, green fluorescence protein, for example, if you attach a green fluorescent protein to the Cas9 nuclease and it binds to the complementary sequence, that is a reporter assay that shows that the gene that you're targeting is present in the organism. So it's not so much as a mutation application, but it's more on other applications of CRISPR-Cas9, which makes it such a, like I said earlier, such a versatile system.
The one that normally used for the diagnostics, right?
Yes, exactly. Yes, this is more your thing, Dr. Amimah.
Yeah, it's now coming. The diagnostics technology is more coming. It's revolutionized. So now they are applying CRISPR-Cas9 even for diagnostic purposes. Okay, so this is the last question for Dr. Adli. Dr. Adli mentioned that we can also add in repair template for knock-in method. Does that consider as introducing foreign DNA like what GMO is?
Yes, it does, ultimately. GMO is just a general term for genetically modifying organism. And CRISPR-Cas9 is one of the techniques that can be used to achieve that goal. So yes, the answer is yes. Whether it's a plant thing, and then I know GMO is more attached to plant and food industries, but it should actually be applied to every genetically modified organism. And I don't know how that has been so synonym to food and plant industries. But again, the regulation is still developing, I think, as we go along, because this is only recently recognized in the Nobel Prize ceremony. So people are starting to look at the potential, and surely all the regulations will follow.
So, is there any question from the floor before we end the session? So that's all the questions that I have. There's one just received. I am studying 40 resistant genes for BLB disease in rice. Any tips to choose selectively for preparation and further experiment steps in case I want to apply CRISPR-Cas? Maybe this question is relevant to Dr. Asyraf.
Right. Okay. So in the case of BLB, the bacterial leaf blight in rice disease, the target sequence, so the target genes might be the susceptibility genes. So the genes in rice plant that promote the growth of this bacteria during the infection. So you might want to target these susceptibility genes and knocking out and hoping that when this gene is not expressed and not absent anymore in the rice, then the bacteria will be able to infect the rice plant, and you expect to see enhanced resistance to this disease. So the target gene, you might want to look at the literature in the previous study, some of the candidate gene that you can target. So for CRISPR-Cas9 for gene knockout, this is one of the limitations. So you have to have prior knowledge on the gene that you want to target. So if you don't have any target, then you can you're required to do some fundamental study first by trying to look for candidate gene that might be act as a susceptibility gene in rice, and this can be done, for example, by looking at the RNA sequencing method, where you sequence all the RNA and then see which gene is upregulated, which genes are downregulated during infection, and then from there you can get some data and idea which one you want to target for CRISPR-Cas9.
So I think we are getting close to the end of our time together. So thank you for both of you, Dr. Adli and Dr. Asyraf. So I hope the participants and the audience today enjoy our webinar, and keep watching our next upcoming webinars. So for your information, there will be a second series of webinar on CRISPR-Cas9, and hopefully during that time we can do more on hands-on for our upcoming series. So stay tuned. Don't forget to like our Facebook page, Instagram and website, as we will keep you updating with the upcoming seminars through these platforms. So you also can rewatch this webinar again and again and again through our YouTube channel. So we hope to see you again in our next webinar. So thank you so much. Thank you everyone. Assalamualaikum.